At this year's meeting, we reported the end of phase 1 results for the KTE-C19 CAR T-cell trial in adults with relapsed and refractory acute lymphoblastic leukemia. Acute lymphoblastic leukemia remains an unmet need. The majority of adult patients will relapse, and when they do relapse, the outcomes are poor. So this phase 1 was launched specifically for patients with primary refractory disease or those who'd relapsed after 2 or more lines of therapy. The KTE-C19 is very similar to the CAR T-cell therapy that's been approved in diffuse large B-cell lymphoma. It utilizes the same murine anti-CD19 scFv and the same CD28 co-stimulatory domain. Where it's different is we isolate T-cells prior to viral transduction and manufacture so that we can avoid transduction of the blasts. We overall apheresed 54 patients and ultimately treated 45. Infection was the most common reason why patients didn't proceed from the apheresis to the treatment with KTE-C19. For the 45 that we did enroll, as perhaps expected, they were a heavily refractory population. 68% had 3 or more lines of therapy, including 42% who had prior blinatumomab. We had reported at the previous meeting that blinatumomab doesn't appear to be associated with inferior response to KTE-C19. About 30% had prior transplant, and around 37% were primary refractory. These patients had very high blast bone marrow burdens when they came onto the study. The median was around 60%, so in fact, the study did not allow for the treatment of MRD. Patients had to have greater than 5% marrow blast just to be able to enroll. Of the 45 patients that we treated, we reported on safety data at this year's meeting, and then we have efficacy data for 41 of the 45, simply because of our data cutoff time point. The safety, we have no new safety signals. We saw 2 patients with a grade 5 attributable mortality, one with multi-organ failure in the setting of CRS. This patient was treated at the highest cell dose, so I should rewind and say that we explored 3 different doses for this trial. 2 times 10 to the 6, 1 times 10 to the 6, and 0.5 times 10 to the 6 cells per kilogram. We ultimately settled on the 1 times 10 to the 6 cell per kilo dose, and I'll come back to that in a moment. So we saw 2 deaths on the study. One was at the 2 times 10 to the 6 cell dose in the setting of CRS and multi-organ failure, and the other one was at the 0.5 times 10 to the 6 cell per kilo dose, where the patient passed away of a stroke, again in the setting of CRS and neurotoxicity. Both of these have been previously reported, so we're delighted to say that the overall mortality amongst the 45 patients that we treated remains quite low. It's 4% for the trial and is in line with what we've seen in diffuse large B-cell lymphoma and the other studies of CAR T-cell therapy that have been conducted to date. In terms of safety, we still do see high rates of CRS and neurotoxicity. If you look across the entire trial, it was around 30% for grade 3 and above CRS, and it was around 38% for grade 3 and above neurotoxicity. We wanted to figure out whether we could do better. We were already seeing very encouraging activity and an improvement in the overall CRS rate as we went from 2 to 1 times 10 to the 6 cell per kilogram. We wanted to see whether we could do better in the group treated at 1 times 10 to the 6 cells per kilo. So we developed a modified toxicity algorithm. Specifically, we approached these patients with steroids at the onset of grade 2 neurotoxicity rather than waiting for grade 3 neurotoxicity to emerge. And we also restricted tocilizumab for the treatment of CRS. So we're no longer recommending to investigators, if they have isolated neurotoxicity, try tocilizumab first. We said, nope, just go straight to dexamethasone, again, if they're meeting that grade 2 cutoff. What we saw was dramatic. If we restrict our analysis just those patients treated at the 1 times 10 to the 6 cell dose, previously we saw around 57% grade 3 toxicity, and I think it was 8% grade 4 neurotoxicity. With the modified treatment approach, we saw no grade 4 toxicity and grade 3 toxicity in 11%, which equates to 1 patient out of that group. Not only do we see a decrease in toxicity, we also saw a decrease in the duration of that toxicity. So the median went from 20.5 days to 11 days in those patients who experienced any neurotoxicity. So we were excited because we feel like we've got a dose now that brings down our CRS rates and a toxicity algorithm that brings down our neurotoxicity rates. What about efficacy? Because again, although it's a phase 1 trial and this is a secondary endpoint, I think it still deserves some attention because we just want to understand this is going to be feasible as we move forward into the phase 2 clinical trial. So looking across all 3 doses, we saw very high rates of CR/CRi, all of which were MRD negative. So in total, 68% achieved a CR/CRi, again, all of which were MRD negative. If we focus on our recommended phase 2 dose, the 1 × 10⁶ cell per kilo dose, 84% achieved a CR/CRi, again, all of which are MRD negative. And more importantly, these responses were consistent across all of our key covariates. Prior transplant didn't affect this. If they were primary refractory, didn't affect this endpoint. And so we're very encouraged by this response rate. We also did present durability of response. Again, this is preliminary because this is a phase 1 trial, but we're very encouraged. For this highly refractory patient population, the median duration of remission at the 1 × 10⁶ cell per kilo dose was 12.9 months when censored for transplant. Interestingly, when we no longer censor for transplant, we don't reach our median duration of response, which begs the question, is this a bridge to transplant? And the answer is I don't know. We transplanted a total of 3 patients at the 1 × 10⁶ cell per kilo dose, and it's really too few patients to make any meaningful conclusions to say, yes, this is the standard we should be taking, or no, this isn't the standard we should be taking. I think for now it's going to be an individualized decision, and I really like some of the data that's emerging around the role of MRD, or in some cases very proliferative leukemias that may be associated with high LDH or low platelet count as the Hutch has developed. So there may be surrogates that we can use to say, hey, this is a higher risk patient where we want to think about transplant in this setting, rather than approaching it as a one-size-fits-all. So to summarize very quickly, we treated 45 patients, including 23 at the 1 × 10⁶ cell per kilo dose. We have very encouraging efficacy, very encouraging safety. We're already about 2/3 the way into our phase 2, and we're hopeful we'll have some good news to share in the near future.